In neuroscience, spikes are action potentials, or the firing of neurons, while the term dark neurons indicates just how rare these events really are in the human brain. That is, when we picture a brain, we sometimes imagine it flooded with action potentials, 100 billion neurons all activating nearly simultaneously. The reality is that there are far fewer neurons active at any given time. Dr. Mark Humphries, the author of the 2021 book, The Spike, An Epic Journey Through the Brain in 2.1 Seconds, has dubbed this phenomenon the dark neuron problem. In this episode of the Thinking Tools podcast, I speak with Dr. Mark Humphries about this and a few other ideas from The Spike. Mark Humphries is a computational neuroscientist and writer. His lab at the University of Nottingham, UK, researches how groups of neurons collectively encode information about the past, present, or future in order to guide behavior. He writes about systems neuroscience for a broad audience at medium.com. By the way, if you're new to this channel, please hit the like and subscribe buttons, and don't forget to sign up for our weekly video newsletter. The link is in the caption, or you can go to senseofmindshow.com/newsletter. Now, without further ado, my conversation with Mark Humphries. Okay, so I will have read your full bio for the audience, but maybe you can, before we jump into the spike and neuroscience and all that, maybe you can just give a background on yourself and why you decided to study computational neuroscience, and maybe just what that field is for people who don't know.
Yeah, okay, sure. Let's start with what it is. Yeah, it's a good question. So I when writing about myself, I often decide not to write down computational neuroscientist because either it sounds really fancy and complicated or no one knows what it is. So it's just the application of mathematics and computer modeling to the problem of how the brain works. So it ranges from the people who work in math departments who like to write everything down with pencil and paper and like explicit equations and solve them, and they want to apply those formalisms to studying the brain, all the way up to people who do, well, like me, who do simulation, who build big simulations of bits of the brain to find out how it works, much like a weather forecaster simulates chunks of the weather. Lots and lots of equations being solved by a computer that spits out outputs that we pretend are things to do with the brain. So that's computational neuroscience. It's where all the theory part sits. Obviously, I never even planned to become a computational neuroscientist, because no one plans to become a computational neuroscientist, because that doesn't exist until you're sort of at PhD level. Mm-hmm. There are no undergraduate degrees in it. There are only a handful of master's programs in it, at least in the UK. So I started out wanting to be a chemist originally. So I had interest in chemical engineering and was going to go to do chemical engineering as a degree. And then the last couple of years of high school here, I really struggled with the chemistry to get motivated with it, and I wanted to find another degree course to go on. And it just so happens that pretty much next to chemical engineering and the undergraduate handbook was cognitive science, which was essentially the study of how the brain functions and the mind works. So that sounded really cool. There's a bit of artificial intelligence in there, a bit of language stuff, some philosophy, some stats, the whole melting pot. So I applied for that. Ended up doing it at University of Sheffield, where there was a really strong computational neuroscience team teaching on that cognitive science program. So we got fed lots and lots of sort of a very early age, fed lots of information about what computational neuroscience is and how you do it. So I did my PhD in it and carried on from there.
Nice. Yeah, I would say that the the chemistry background comes through in the Spike, at least, which now I kind of want to get into that. That's your book, The Spike, and I'll say it's a really just unique take on neuroscience, on trying to explain how the brain works. And in this interview, I want to focus on one area in the book in particular, on the dark neuron problem, but I want to return to that in just a minute. Before we get to that, maybe you can just give like a brief summary of what the book is all about, or some of your main arguments, just so people kind of have an idea going into it, because we'll kind of dive into more specific topics in just a second.
Sure, yeah, okay. So I guess, the book is really sort of three journeys in one. Right? So at one level, it's it's a narrative journey of following a single spike, a single action potential, all the way from when they're first evoked, when light hits your eye, through a journey through the brain, through various regions of the brain, like a big tour, to understand what various regions of the brain are doing and what role they're playing in a very simple act. In this case of seeing a cookie in a box and reaching a hand out to try and grab it. So you've got this sightseeing tour through the brain. But the whole reason I structured around the sightseeing tour was because as you go on that journey, the reader is being introduced to more and more detailed and deep concepts about what these action potentials, these spikes are, how they're made, what they mean when they're sent. So this journey takes them deeper and deeper and deeper into the science of this level of the brain. And then the third journey, because it goes deeper and deeper and deeper, it turns out being a journey deep into the sort of the current cutting edge of neuroscience, of how we can record these things and what we can do to manipulate them. And as you say, I wanted to write this book because there is really no like popular science book that treats either of those two latter journeys, either of how spikes work and the fact that they are fundamental to how the brain makes you do anything, and no one explains that. And there is nothing at all on the fact that we're currently in what many consider to be the golden age of systems neuroscience, where we can record hundreds or thousands of neurons individually at the same time, and we can manipulate them using light and drugs and turn them on or off at will, and even trace where most of their wires go. So we have this extraordinary sort of accumulation of knowledge at the moment, and it was a good chance to get it out there.
Yeah, and it's really amazing, the kind of the level of detail, but also the breadth of the book, because as you say, you're really following an individual action potential, and it's centered on an individual person doing something, as you said, so it has to have a wide breadth. But so that being said, the thing in your book that really just fascinated me, and I couldn't believe that I hadn't heard it before, was this idea of the dark neuron problem. And so maybe you can describe what the dark neuron problem is, and then we can kind of talk about the type 1 and type 2 dark neurons and get a little more into that. But perhaps how it was discovered, or just an overview of the problem in general.
Yeah, okay. So the short version of the dark neuron problem is that there are far fewer neurons that are active than you would think, given any sort of passing knowledge of neuroscience. And this was even only revealed recently to neuroscientists, let alone to the sort of more general audience, because up until about 25 years ago, the only way we had of recording neurons was to lower an electrode into the brain. So when you lower an electrode, you can't see what they're recording from. So the only way you can tell you've found a neuron to record from is because the electrode is picking up the signal from the neuron. So that meant, obviously, if there were any neurons that were not active, they would be invisible to the electrode. So we read many tens of thousands of papers that reported on we load electrodes into, motor cortex of rat X or prefrontal cortex of mouse Y, and we found these neurons that responded to this thing, or arm movement, or to a maze turn, or a piece of fruit or something. So great. And then the last sort of 25 years, the ability to image neurons came along, the ability to essentially stare at them through a high resolution video camera and have them fluoresce when they were active. So, because now they're lighting up when they're active and you're staring at them, you can see all the neurons that are in the scope or whatever sort of microscope you're using. You can see they're there. You can also see that they're not firing. So you can see all these neurons that are not active at all. And that really confirmed that there must be a lot of neurons that when you're looking at a chunk of brain for a long chunk of time, like 10, 20, 30 minutes, there's a whole bunch of neurons that are barely sending any spikes at all. And then in retrospect, that had to be true. People have long worked out that when you put an electrode into the brain, of course, it's going to should pick up the activity of all the neurons that are around it. And if it did that, then you should pick up about 100 to 200 neurons on a single electrode. But as you're only ever picking up 1 or 2, that implied that actually all the other neurons around it were not firing. But as ever, that's that sort of reverse inference thing of going, well, they ought to be there, but they're not. So that's on sort of slightly shaky ground until someone can actually look at the neurons and go, yep, they're definitely not firing.
Yeah, and it's so counterintuitive given what the pictures of fMRI scans that most people have seen look like, oh, there's this big chunk of brain that's lit up all red and bright, and it looks like, oh, that must be that whole area, millions of neurons active at once. But what this research shows is, no, it's a relative few. And in fact, you say at one point that it's 10% of cortical neurons produce half of all spikes. And so that, I mean, that's just amazing.
Yeah, that seems to be pretty robust across studies, and even then, that's estimates coming from again studies using electrodes, but lots of them, and recording for a long time, so they can pick up neurons that are firing only very infrequently, say once every couple of minutes. So the true number is likely to be even worse than that, like to be even fewer than, or I think 10% will give even more of the spikes rather. And yeah, so yes, as I said in the book, it's kind of it is this kind of long tail thing where there's a small number of neurons that are taking care of almost all the spikes that are being sent in a given area. But that, of course, obviously in the book we talk about how that might be slightly misleading way of thinking about what these neurons are doing. So all this 90% who are sending only 10% of the spikes. Of course, that's not many of the spikes, but there's a lot of neurons. So if they're sending them collectively, of course, that's still a lot of information being sent, just not very often.
So is that your favorite hypothesis for what they're doing? Because in the book you mention a few different ideas about how to explain this problem, which you label the dull world, the reserve army, and then what you just said is that they're spending spikes, but just not very often. Could you explain those different ideas and which one you think is most likely, or which combination?
Yeah, sure. So let's start. So I was starting to talk about this idea that this sort of collective activity solution, that the idea that yes, there's many neurons who fire very, very few spikes, but when they do fire spikes, because there's so many of them, obviously anything receiving those spikes is going to likely respond. So they're quite capable of transmitting information. They're just not doing it by sending a lot of spikes individually. So we see an example of this. There's an example of this I think in auditory cortex, where there's this phenomenon called binary spiking, where, although spiking is binary, it's a phenomenon where you play a sound to a rat and you're recording him the first bit of auditory cortex, and the neurons there in layer 2-3 only respond with a single spike to that sound or nothing at all. So you can read across the population and see what particular sound got played, because you just read off the neurons that responded. So you have this, they send information collectively across the spikes. They don't send many, but when they do, that means something very particular. So another option is that, yes, this dull world hypothesis. And that's one that's really been sort of driven by people who look a lot of visual cortex. And they've been very interested in this idea of redundancy, of the fact that you only want neurons to fire when they've got something to say. And you don't want lots of neurons firing together, because we're wasting energy sending information that's already covered by other neurons. So in that view, then what the dark neurons are they're neurons who are just not engaged by whatever is in the world. So the best way to make sure that you'll have a really energy efficient brain is that you have neurons that respond only to very selective things in the world. So neurons that only respond, say, to a particular vertical line at a particular position in the world at a particular thickness. Then that particular vertical line at a position of thickness only ever occurs very occasionally. So they only ever respond very occasionally. So they'll only fire very sparsely. And elaborate that out across the whole of sort of visual cortex, all the neurons that respond to all sorts of different types of little edges and stuff. Then at any given moment, only a handful of them will be firing. So they'll look really sparse. There'll be a lot of dark neurons that won't be firing. So that's the dull world argument, is that we just show an animal really boring pictures. Most of its neurons will do nothing at all. If you manage to put it into a more rich natural environment, then the activity will probably grow, because there's far more complex series of edges and curves and so on to respond to. And yes, And the last one, this hypothesis, this reserve army thing, is just is a speculation about the fact that obviously, particularly for primate brains, we have long lifespans and we have lots and lots of life experiences to learn about, lots of skills to learn, and things we can learn throughout our entire lifetime. So we need to have the sort of neural real estate available to start representing stuff that hasn't been represented before. And perhaps what a bunch of these dark neurons are is this neural real estate. They're being kept alive by occasionally being active, but they're essentially waiting to be recruited for a job. And there's some sort of rough evidence along these lines in hippocampus, where we know in hippocampus you have these place cells, these neurons that only fire when a rat goes in a particular position in a maze. And they seem to be recruited from cells that are very, very sort of low activity. So you can recall a cell when it's wandering around and it walks into a new part of the maze, and suddenly the cell will burst into life as a place cell. So they're sitting there as a reserve set of cells waiting to be recruited to represent place. So, yeah, I mean, I can potentially, of course, all three of them are true, that they don't have to be mutually exclusive. That's the nice thing about science often, right? We can hypothesize a bunch of different stuff, and in the end, it ends up being all of them in some way.
Right.
So because I spent a lot of time looking at this kind of problem of how neurons encode information collectively as a group, I favor that initial hypothesis of they are sending information just fine, just not in a lot of spikes. But obviously I have to admit the other two are plausible. Yeah.
Gotcha. Yeah, that totally makes sense. All right. So backing up just a second. So we talked about that this is true for cortical neurons, and I think you mentioned, or maybe you didn't, but does this also hold for subcortical structures, even down to the brainstem? And if so, what do you make of the kind of 10%, the myth that says that we only ever use 10% of our brains at a time? So subcortical structures, and then that myth.
Yeah. So it's below the cortex, there are the wildly differing sort of amounts of activity in different regions. So there is, for example, the striatum, which is sort of what takes up almost the rest of the forebrain underneath the cortex. So for some scale, so in a rat, the cortex of a rat is about 17 million, 20 million neurons, and its striatum is about 6 million. So it's a very big thing, and it's almost it's more than twice the size of the whole of the hippocampus. But it's also legendarily extremely quiet, even more quiet than cortex. So if you sit in a lab while someone is trying to recall from the striatum, they lower an electrode through the cortex, and you can hear it ticking away on the lab speaker with the spikes going through the electrode, and the tip of the electrode plunges into the striatum, and the speaker is dead silent. So there's millions of neurons in there firing very, 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 very sparsely. And yet, some of the regions the striatum connects to, these tiny structures called the globus pallidus and other regions next to it, they're firing in the rat, they're firing at least 30 spikes per second each. Every neuron you can find is firing these huge numbers of spikes. And indeed, even they fire so often that if you take these neurons out of the animal, put them in a petri dish, they'll keep firing. They don't need input to fire, they'll just keep firing. They're pacemakers, just like the heart pacemaker. So below the cortex, it really depends on the job of the region. Some neurons have these extraordinary constant activity, and other regions are almost completely silent. They're doing very different jobs. Well, of course, none of those quite speak to the 10% myth. Right? So indeed, someone asked me just the other day about that 90%, 10% split of 10% of the neurons firing at least half the number of spikes in cortex, even higher. Is that where we get that 10% myth from? And it's not. It just happens to be a total coincidence that 10% of the neurons fire almost all the spikes. That came from, that's normally traced back to Lashley's work in the 50s, where he tried to work out where memory was stored in a rat by having rats run these really complicated mazes and taking off chunks of their cortex to try and find out when they would fail to solve the maze task by taking this cortex off. And to his frustration, he found that it didn't really matter. As long as he spared at least 10% of the cortex, the rat could solve the task. So it seemed to be a misheard interpretation of that is, 90% of the brain isn't being used. Obviously, that's not, not the case. It was. They could solve this task with only 10% of cortex. But of course, to do more complicated things, particularly social interactions, rats need more of their cortex.
Okay, yeah. Well, that's good, because I've been telling people that's a myth for a long time. So, all right. So, kind of jumping back to fMRI and other brain imaging techniques. In your view, what good is fMRI? What is it really? I mean, it's telling us changes in blood flow to different regions, but as we know, it doesn't necessarily correlate to, if this region has a huge amount of blood flow, it's necessarily got a huge amount of neural activity going. So what is it telling us, and what can we infer from those studies?
Yeah, so I guess, it's a really good question. It's a question that quite a lot of people have grappled with. So fMRI is, on one hand, fMRI is a wonderfully powerful tool, right? It's certainly the best way that we have of looking inside the living human brain while it's doing stuff. But as you say, it has the limitation of what's being recorded essentially is oxygen-rich blood flow, and that's only a loose proxy for the activity of neurons, and even then, it's just the activity of many, many hundreds of thousands or millions of neurons simultaneously. Obviously, it can't get close to the level of the spikes that we're talking about now. So it's many orders of magnitude away. So that really means it's a little bit limited in what it can tell us about how brains work at a fundamental level. And when I'm pressed to say what has fMRI told us, generally I come up with two things. One is, it's obviously the only way that we can really study how spectacularly lateralized the human brain is. So obviously certainly most mammalian species seem to have some kind of imbalance in the left and right hemispheres of the brain about what does different tasks, but obviously in humans that seems to be extreme, not least because our classic fact that language on the left-hand side is not on the right. The fact that we're extremely handed, we tend to be mostly right-handed rather than left. And there are other studies and many other properties of the sort of human mind that when you look in the activity in cortex seem to be lateralized. So see, that's the only way we have of understanding that lateralization, because there's no other way of looking at it properly. And the other main thing that fMRI has told us a lot about is this default mode network. This network of activity that's across the entirety of cortex, and indeed some subcortical regions, that exists only when people are in this resting awake state. So when they're sort of not actively engaged in a task and have sort of in their quiescence sitting there, activity in a whole bunch of regions pops up, which would have been very hard to discover in a rodent or any other more complex mammal, because you wouldn't really tell whether they were resting or not doing something. It's much easier to ask the person to properly relax, and so you can see this thing. And that's been linked to all kinds of interesting stuff, obviously, about the daydreaming and rumination, about the level to which it's obviously all the activity in these regions gets suppressed when you actively engage in a task. And so that's been an interesting sort of thing, certainly discovered first in fMRI, and people have now actually done fMRI studies of primates to find that it's there in monkeys too. But obviously these are very different questions to the sort of things I like to ask of the brain.
Yeah, definitely. So just drilling into that a little bit more, I mean, what do you think if somebody says, well, this area of the brain activated while the participant was doing some task, and therefore it's involved in that task? Is that like a convincing argument to you, or do you think of it like, well, we're not really sure if the neurons are like, what's going on there? Is it just some other physiological change that happens to be correlated with it? How do you think about that?
Yeah, I'd say, yeah, in general, the kind of study which reports we gave participants some tasks to do and area of brain X lit up are not particularly interpretable as insights into how the brain's actually doing stuff. The best kind of studies of this attempt to correlate the activity they're seeing in the fMRI to a particular model of how the task would be solved. So they run a little computational model of, say, how the decision will be made between 2 or 3 options, say, and we'll be looking for activity that corresponds to, say, the error signal in that model. And then when they find it in a particular brain region, then they have evidence that that brain region is either making or processing that error. And in these kinds of, the best kind of these kind of studies are often looking for whether there are different types of error in different types of brain region, implying different computations are happening independently, which itself would be interesting to know about. So this sort of, the much more sort of theory-driven use of fMRI can start teasing apart what's underlying behavior, the sort of thing you then really want to go to an animal model and find out if it actually is what the neurons are actually doing in that region, because it can only tell us, say, there's a greater amount of neural activity in this region related to potentially something in the task. How that's encoded, how that's sent, how that's computed, obviously is invisible. So yeah, there is always a hard limit to what we can know.
Gotcha. Okay. All right. Well, we've talked about type 1 dark neurons, which are the neurons that don't seem to be sending a lot of spikes. Now, there's also this other type that you call type 2 dark neurons. So can you explain what those are and how they're different and how they're discovered?
Yeah, sure. So those are neurons that I wish I'd given a different name to. So obviously, deep into writing the chapter, okay, type 1 dark neurons are the ones that don't barely fire. So these type 2 dark neurons is carrying on the analogy with dark matter. Obviously, is that the idea with these neurons are invisible to the outside world. So the type 1 dark neurons are invisible because they don't respond. The type 2 dark neurons are invisible because they are perfectly active. It's just that they don't seem to be responding to anything happening in the outside world. And we've had reports of these kind of neurons for decades. So all these many sort of electrode-based studies I've talked about, where you lower an electrode into part of the brain to record neurons, they would give the animal a simple task. So they would show it some simple pictures or play it some simple sounds, and they would record a whole bunch of neurons and then report, okay, so about 50 of the 200 neurons we recorded from responded to the pictures. So we're going to talk about them and ignore the 150 that didn't respond to the pictures. And then the more we dig into these larger and larger recordings of lots of neurons recorded simultaneously, the more we're finding that there's lots and lots of neurons that are active, but don't seem to be particularly caring about whatever the experimenter is doing outside in the world, or what the animal themselves is actually doing about particular arm movements it's making, or paw movements, or whatever. So these type 2 dark neurons are the ones that are perfectly active, just don't seem to be listening to anything that's happening outside. And again, that obviously picks up a whole bunch of questions about what they're for now. And one is the sort of simplest thing they might be for is just to make things go faster. So the mere fact that they're active means that the neurons that are receiving their spikes are themselves more likely to send spikes. So then when something happens in the outside world, everything can respond faster because everything's already primed to respond, because there's all these spikes sloshing about that are making neurons close to the ability to fire. So a little input comes from the outside and suddenly they can fire. So that works. The models have shown that will work just fine. That it's perfectly plausible. It just seems to be an extraordinary waste of energy to be sending all these spikes all around cortex just in case something happens, so you can respond faster. So the other sort of main insight about what they might be is that they're actually signatures of sort of the underlying constant sort of dynamics that are happening in cortex, the constant thing that is generating all these spikes. And then that constant activity is there because what it's there for is to do important computational stuff that the input from the outside world is then added to. So there's ongoing computation. So what we're looking at when we're seeing these type 2 dark neurons everywhere is possibly that we're looking at the signatures of ongoing computations in a whole bunch of brain regions that we just don't understand, because normally we'd like to relate stuff to the outside world. It's very difficult to relate what neurons are doing. We can't manipulate how they fire.
Right, right. And if you were to speculate, what kinds of computations are they doing? What might that be contributing to?
So, obviously in the book, I talk quite a lot about how this ongoing activity might be related to these variety of predictive processing accounts of how the brain works. So this is particularly popular with, again, with the sort of the vision side of things, perception, this idea that what much of the activity in the brain is doing, in the sensory systems at least, is predicting the inputs that will come from the outside world, so that your higher areas of your visual system, for example, are predicting the objects and colors that you will be seeing next, on the basis of the fact that the visual world doesn't change very quickly, so it's easy to predict what should be coming next. So that way you don't have to process every single thing in your field of view. You just have to process what's changed, and all the actual input coming from the outside world through your eye is telling your brain is what has changed, what doesn't meet that prediction. And to do that, of course, you would need a huge amount of activity sloshing around in the brain. So when we see all these type 2 dark neurons, all this ongoing activity, it makes sense then that if those accounts are true, that's what that activity is. It is the predictive processing. It is the generating the predictions about where things should be and what shape they should have and what color they would be and what texture they would be. And that's all being fed down to the early visual parts of the visual system right at the back, so that when the input comes from the eye, it can either be matched and we know it's correct, or it can change and update that prediction.
Yeah. So I think, so would that imply that our moment-to-moment perception, whether visual or auditory or whatever, is really the prediction, and then that prediction just being adjusted by the inputs? Is that a good way of thinking about it?
Yeah, certainly quite a few people think about it that way. Yeah. So Anil Seth, for example, calls this the controlled hallucination hypothesis. The idea that, yes, you're essentially, your activity in your brain is, for visual perception, is essentially hallucinating stuff, and that it's making these predictions, but they're sort of in a controlled way. It's obviously making predictions based on what you already know about the world, not just free form hallucinations. And then the inputs coming from your eye are then adjusting those to get them closer to reality. So the people who talk about this obviously point to a whole bunch of visual illusions, which suggest, of course, that we come with all sorts of top-down predictions about what the world should look like. Things like the classic illusions where you have a room that's drawn to come out this way, and you have a figure that has 3 figures placed in that room, and the one at the front looks enormous, and the one at the back looks tiny because the room is sloping down, but actually the 3 figures are identical in size. That's because your brain top down predicts that because it's sloping, the figures must be getting smaller. So it just adjusts your incoming sensory input, which of course is telling you exactly where the edges are. It's saying they're there and there and there and there. So it must be adjusting that to tell you that they're different sizes. And indeed, there's some evidence from people who suffer from auditory hallucinations. So people with various forms of psychosis, that when you induce auditory hallucinations in healthy individuals, the way it seems to be induced is because they're sort of, if you back up a second. So this experiment is really cool. So we have the auditory hallucination experiment. So what we're doing is you play someone a barely perceptible sound and ask them whether they heard it or not. So yes or no. And of course, because it's barely perceptible, on a various number of trials, you don't play the sound, and they report hearing it anyway because they're listening so hard that they think they hear the sound. And so that means you can induce that kind of hallucination in a perfectly ordinary person who thinks they've heard a sound. And then again, this is one other quite cool use of fMRI, is you can go and look in their brains at that point, obviously, and see that the activity in prefrontal cortex was as though they were predicting there was a sound there and nothing happening in auditory cortex. So it seems to be that we fed a computational model to this. What they're doing is they're listening too much to the prediction rather than the sensory information.
Wow. Wow. So there's the prediction can sometimes even override the signals coming in from the outside world.
Yeah, and that's the reason this talking about this particular paper is because they tested this with people who have psychosis, have auditory hallucinations, even with that just normally. Testing this experiment, of course, they hallucinated the missing sound a lot more, which suggests that indeed they overweight their prior experience. They listen too much to what the predictions. And of course, if the predictions then are not constrained by reality because they're predicting all the time that this stuff is there and it isn't, then perhaps one of the suggestions about what's happening in hallucinations in psychosis and schizophrenia is that the incoming information is not correcting the prediction. So the predictions are the hallucinations.
Yeah, and that would seem to relate to maybe to dreams. If dreams are just internal brain activity unconstrained by any kind of sensory information, and I think it's the case that the prefrontal cortex kind of quiets down during dreaming sleep, and maybe that's just allowing these predictions to run wild. I don't know. I'm completely speculating here, obviously. But.
Sure. Yeah. I mean, I think perfectly plausible. Yes. I mean, I think that's partly when people experimented with deep neural networks dreaming by essentially turning off the error feedback and just letting it respond constantly to an ongoing set of inputs. Then for neural networks that are just generative models of, say, artwork, then they produce really quite fantastical mishmashes of stuff they've been trained on while they are dreaming, because they're just all the top-down stuff from the inputs is from for what they're trained on is generating output, but they're having no error feedback to correct what's happening.
Wow, it's just fascinating. I mean, in the book you mentioned that well, this is kind of a question I wanted to get to a little bit later, but I think actually it makes sense to talk about it now. So I've been asking all my guests to give me an elevator pitch answer to the question of how the brain works, which I know is kind of ridiculously general and huge, but you give a nice little elevator pitch answer at some point in the book. I'll just quote you really quick. You say, quote, "I'm proffering here a simple but rather radical model of how your brain works. Spikes from the outside world adjust to spontaneous activity, and these adjustments are the messages they carry." So like, that's what we've just been talking about, but I think it could slip by people that this may be a good idea of thinking about how the brain works in general. Is that how you would characterize that?
Yeah, I would. So I think, so in fact, a bit independently of this whole predictive processing idea, obviously that's an idea that's come from sort of top down of people looking at how humans seem to process information and going, this really looks like we're doing this predictive processing thing. So see where the book builds up from the other direction, it says, look, we build up all this knowledge of how spikes work and how we generate all these spikes, and the fact that we would expect that in a large enough brain with enough neurons connected together, they would just spontaneously make a lot of spikes, irrespective of what's happening in the outside world. Which means that really the brain is a whole set of dynamical systems that are just created by these neurons that just feed back to each other and create all sorts of wonderful patterns of ongoing activity. So in that view of the world, what's happening, the input to the brain, wherever it comes from, all it's doing is essentially nudging those dynamical systems into different states. So it's just changing that ongoing activity into a different pattern. So rather than the incoming information creating particular spike patterns that mean, there's an edge, there's a sound, I'm thinking of crisps, whatever, that or those, they don't actually mean that. What they're doing is they're adjusting ongoing activity, and that ongoing activity means that. So the meaning's there in these ongoing spikes. And then that fits with the predictive processing accounts, but it's broader than that. It's about, we see these kind of dynamics in motor cortex when animals move their arms, and all kinds of regions of the brain. So yeah, it's as an elevator pitch theory of the brain, it's I normally write down something like the brain is a big bag of cells wired together. We'd expect the whole thing to be one gigantic dynamical system, just for which every input to it is just pushing that dynamical system into a different state, if you like.
Wow. Yeah. All right. Well, related to that question, what would you say are some of the biggest misconceptions about how the brain works?
Oh, I think we've covered quite a lot of those already, haven't we? We've covered the 10% of the brain argument, and the fact that neurons are always active, and fMRI, what it can see. What else is there? Well, I guess there's a lot of misconceptions, I guess, about how much cortex does. So the fact that we know so much about cortex is really kind of an accident of the fact that because we're humans, we're interested in what in sort of human processes most, and because we're visual creatures, we want to understand vision the most. So obviously, most of the complex vision processing happens in cortex, so we have to record from cortex. Fine. But of course, there's a whole other chunk of brain to be worried about other than cortex. And as we kind of touched on earlier, rats can do perfectly well without cortex. They don't really, they do lots of advanced processing with it, but there's a whole classic series of studies from the 80s and early 90s from Ian Wishaw, where they aspirated, so vacuumed off the cortex entirely from the outside of various rat brains and observed them for months on end. And all they could note that they weren't very good at was social interactions, because they no longer had a prefrontal cortex to suppress some of the more sort of basic urges. And they couldn't trim their toenails. So there's these great pictures of these enormous rat nails that weren't trimmed because they couldn't coordinate biting them properly. And other than that, there were very few obvious issues that they had. So taken to an extreme, obviously that like my old boss Pete Redgrave used to refer to the cortex as a blanket that keeps the rest of the brain warm. From that point of view, because it's just a lovely thin layer of cells that just keeps the heat inside in the middle, keeping all the other neurons nice and operational. So because there's a tendency obviously to view the cortex as this seat of all the higher functions, and the rest of the brain is doing some kind of plumbing work, right? The difficult stuff. But really, there is no sort of separation. Yet, sure, in humans the cortex is quite elaborated, but so are huge chunks of the rest of the brain. There are, the example of the striatum. The striatum was also a huge structure in the human brain, and damaging it in any way causes all kinds of disorders, so Parkinson's and Huntington's and Tourette's and OCD and all kinds of things. So it's really best to think of the brain as a whole, when a brain as a holistic thing, and there isn't. And so this misconception, there's this kind of cortex that separates from the rest. It's a strong one, particularly people who still cling to this idea that simpler animals don't have cortex, that there's kind of like a 3-level brain thing. There's like a reptilian brainstem thing, and then there's like a simple mammal midbrain thing, and then there's a like advanced creature cortex thing. There is no vertebrate that doesn't have something that looks like cortex. Even the lamprey, which is like a tube of teeth, basically, which diverged from us about 350 million years ago, it has a little tiny neural crest thing sitting on top of its midbrain, which has got a few cortex-like cells. It's not layers, but it's a thing that's there. So, so the really best to think of not the cortex as some kind of seat of higher function, but it obviously integrates with the rest of the brain to produce whatever it produces.
Right. Yeah. No, that makes a lot of sense. I had never heard that about the rat study. But I guess we should just mention that there are cases of, at least I've read about people born without a cortex, and they are just profoundly disabled. So it may be, I mean, do you think it's probably more important for normal human function than maybe for rats? Is that, would you agree with that?
I think, well, see, any genetic abnormality that's so severe that there is poor development of the cortex, or indeed some of these cases where huge ventricles, so all the brain is pushed right to the extreme. Of course, that also presumes there must be damage, development abnormalities in many other places too in the brain. So it'd be hard to say this is a problem just because cortex is damaged. But it's certainly possible, of course, that because if cortex doesn't develop properly, then presumably lots of chunks of the brain underneath don't develop properly either, because they're missing those inputs from all those cortical neurons that are needed. And lots of cortex is the part that develops most in the early years of life. There's some studies even suggesting that our prefrontal cortex doesn't stop maturing until we're in our early 20s. So, as you say, it's also equally possible that in humans, just missing a cortex because it's so integral to coordinating all the many advanced functions that we do means that we're reduced to a state where, we're even more sort of limited than a rat missing their cortex, because the rat's cortex isn't quite so elaborated. It doesn't quite have taken over so much of the stuff that it does.
All right. Yeah, that does make sense. So as you mentioned, we don't necessarily know as much about what the subcortical structures are doing, only because the cortex is relatively more visible, easier to access. And in the final chapter, I believe, you start talking about some of the technologies that are emerging in the last few years and that are emerging now that will allow us to understand the brain at a deeper level by focusing on spikes, on what neurons are actually doing. I was wondering if you can talk a little bit about some of those techniques, maybe about some of the older techniques and how they've evolved. Leave that to you.
Yeah, sure. So where we start? Okay, so with this, we talked a lot about recording with electrodes. So obviously, historically, literally just bits of wire that get lowered into brains. Obviously, electrodes have evolved into an sort of highly finessed state now. So there's everyone is very excited about these so-called Neuropixels probes that have been available now for 4 or 5 years. And they're electrodes that are extremely long and thin, and they have on them the first generation had on about 400 contact points. So 400 little, essentially individual electrode recording points on this whole shaft. And a shaft that's long enough that if you put it into a mouse brain, it would go essentially from the top of cortex most of the way through its midbrain to its brainstem. It became this way. So not only can it record many, many, many neurons at once, it can also do so over a very large span of the brain. And one thing this was immediately cool for is that because it just happened, because a lot of people using it were really interested in cortex, but they couldn't just put the tip in, they had to put the whole thing in. So they would look first at those top electrodes that were in cortex, and then they could look along the rest of the shaft and see that actually all these neurons in these weird areas we've never looked at before are also responding to these tasks. So crazy areas like the zona incerta and the central gray, things that are barely even registered on most neuroscientists' name lists, suddenly have these neurons in them that seem to respond to stuff. So that means that these new electrodes have been fantastic in immediately breaking open this sort of cortical chauvinism thing of just looking at cortex, because they happen to record from lots of neurons elsewhere anyway, so they might as well look at them. So that's really feeding into this whole brain as a big bag of cells idea, that these signals are everywhere. So it's very difficult to say this bit of cortex processes the decision to make a movement when you can see cells down in the brainstem that are processing it at exactly the same time. So that's a technology that's just come available. That's still developing. The new versions of that are just about to come out. I think will have come out even more dense in the recording sites. The thing I'm most excited about, I think I'll talk about in the book, is the prospects for voltage imaging. So to explain that, where do we start? So earlier we talked about imaging, about the idea that we can record neurons essentially with a video camera by pointing a video camera down a microscope and filming neurons as they're active. As I said, I talked about the fact that we film them while they are fluorescing, so they're giving off light while they're being active. And for most of those types of studies, what they're actually looking at is not spiking all the neurons' voltage, they're looking at the calcium inside the neuron. And they're doing that because it's much easier to get a protein, a fluorescent protein that binds to calcium, so that when the calcium inside the cell increases, so the fluorescence of the protein increases, and you can see the light level change through your video camera, your microscope. So you can pick up the light going up and down. And that's wonderful. And calcium imaging has now been used for about 25 years. And you can do it at obviously high. You can pan out and look at hundreds of cells. You can go right into an individual dendrite and look at a tiny individual synapse and see the calcium signals moving around. Fantastic. Well, see, what we're thinking about here is talking about recording hundreds of neurons at the same time, so looking at hundreds of them. Problem with calcium though is it's really slow. Obviously, it's related to the spikes that are being sent, but it's not quite the spikes. So every spike that's sent, there'll be a bump of calcium, but it goes up slow and it comes down even more slowly. You can't really see individual spikes. You can say, yes, it's definitely sending spikes, but we don't really know quite how many or exactly when. So what you'd ideally want then is you'd want to be able to find a fluorescent protein, something that will give off light when the thing it binds to increases. And although we've had them about 30 years, just a couple of years ago, a whole bunch of new proteins appeared that could actually bind to the body of the neuron and respond to its voltage rather than to the calcium. So respond directly to the voltage going up and down that will create the spike, and then see the spike as it's released. So then you can now film hundreds of neurons in theory and see from the fluorescence of the light coming from each neuron, the voltage of that neuron and when it spikes. So the current state of the art is the biggest recordings are about 40 neurons, I think, in a rodent brain. And they can definitely see the spikes, and very occasionally see something that's before the spikes, but obviously the noise is huge. And one of the reasons it's so difficult is, so when you recall from calcium, obviously the cell body is a three-dimensional thing, right? It's a big sphere, and calcium fills the whole of the middle. So when you put the sensor in, it's filling the whole of the cell body and it's fluorescing away, and it's quite a big signal. But voltage is just happening on the outside. It's happening on the shell of the skin. So the sensor is only on the skin. So it's obviously exponentially less light is being given off when it fluoresces. So it's much harder to detect, to get a lot more noise. And also in the cell body, these cells in mammalian cortex are tiny. So a mouse pyramidal neuron in cortex, the biggest neuron in cortex, its cell body is only about 10 microns across. So the signal was absolutely minute. So the fact we now have these sensors that can detect this stuff at all is amazing. So what we'd really love is that technology keeps getting better and better and better, so that the ability to detect the voltage linked to fluorescence gets better and better. So you get a higher signal to noise ratio. You can really pick up when that voltage is changing and do it across hundreds of neurons and have it last longer than about 5 minutes, which is the maximum it does at the moment, because there's so few of it that every time it fluoresces, it decays a little and it'll decay and essentially die off over a short period of time. But the reason we really want this is because it's the best of both worlds. You can see the neurons and see the spikes, which you can either do with electrodes or calcium imaging. So it's both of them put together essentially. So should it become possible to do this at scale, then that would be the default tool really for doing this kind of neuroscience. But yeah, we'll wait and see.
Wow. Yeah. Yeah. I can see we're starting to near our time here. So yeah, I guess just a couple final questions for you, a little bit more general. So if somebody asked you, I can only read one book about the brain, and I've been saying, well, preferably not a textbook, but what would you recommend to somebody if they could only read one book about the brain?
Other than mine, obviously.
Well, that one would be number one, of course.
So it's a really good question, because obviously one of the reasons I wanted to write this book is because it's very hard to find really accurate and well explained and interesting books about how the brain actually works. It's very easy to find sort of very popular science, sort of almost self-help books about how dopamine works, which are full of speculations about how dopamine is for reward, and maybe it's got us enslaved to our mobile phones, our notifications, and social media and stuff, without any basis in science. So for people who are interested in a really in a nice, really well researched sort of historical narrative of neuroscience, of what we know and how we got where we are, then Matthew Cobb's book from a couple of years ago, The Idea of the Brain, is sort of his magnum opus of a beautifully researched history of how we know what we know now, and sort of the modern, where we are right now, sort of more elaborate version of what I've been talking about, and then what might be happening in the future. So he draws on all kinds of strands of neuroscience research, and nicely chunks up the kind of the modern section into sort of biology, chemistry, and physics, to kind of explain how those three fields have filtered in to neuroscience, to make it this massive melting pot of every single discipline under the sun, essentially, inside science is doing neuroscience in some way or another.
Well, that's great. I've been seeing that one floating around, but I haven't picked it up yet, so I'll definitely check that out. And then finally, in addition to your book, you also write a blog on Medium about similar topics. So I'm wondering, what lessons would you have for somebody who wants to communicate neuroscience or science in general to the general audience, to the general public?
Well, short version, I guess, is it's really good to follow the standard advice that your reader is intelligent but doesn't know the words. So they will understand almost any complicated concept as long as it's explained in accessible language, in this case, accessible English. Right? So, and it's very easy as a scientist to not do that, because even the most innocuous words you think are sort of not technical are actually technical. So, for example, scientists routinely talk about stimuli, and everyone knows exactly what they mean. But to a person who doesn't do experiments, which is almost everybody, they probably don't realize that stimuli always refers to something being shown to a person or an animal on a screen normally, of course, or played as a sound. It is the thing that is being presented to the subject in order to get a response. It's not just any arbitrary stuff that's in the world. So yeah, it's very easy to be tripped up by the language. And obviously, hand in hand that with goes accuracy, right? It's very hard. It's what I try and strive for, particularly in the blog writing, is to convey these complicated ideas accurately. So not being tempted to wander off into deep speculation about, oh, this must mean we can cure depression in a couple of years' time, when I'm writing about new research on how ketamine works deep in the brain. Because it's in many respects, it's more interesting to be able to convey properly the limitations of what we can know of saying, so why doesn't this mean that we're going to have a cure in like 10 years time? Because this is just explain that this is just a tiny dot in the map of this gigantic space we have to explore. But this is really cool, because this is the first step on that map. We've stepped into this region now, and there's a whole area to explore. That's going to be fantastic. Anything else? So accessibility, yeah. Accuracy. Entertainment helps, right? So it's quite a bit. It's kind of essential that the reader knows why they're reading it. So I always try and as you can see in almost all the best popular science writing, is that the reader is told what they're going to be told right at the start. So just like you have in any sort of decent length newspaper article, the lead is going to explain what you're going to be told, so that you already have a framework for what it is you're going to be reading about. Which is quite hard for scientists to switch into, because scientists, obviously scientific papers are not written that way. They're written as an accumulation of results to come to a conclusion that this is so, use this long linear argument of laying out all the sort of background stuff and then these results and then come to a conclusion. Whereas when you're writing for a popular audience, you just start with the conclusion and explain how it got there. So yeah.
That's great. That's great. Accessibility, accuracy, entertainment. Good stuff. All right. Well, we've reached our time. Thank you so much for talking to me today, Mark. It's been a real pleasure, and I really appreciate you just answering all my questions.
That's great. Thank you, Andrew. That was cool.
Thank you. Okay, well, that is it. Thank you so much for listening or watching this episode of the Thinking Tools podcast from Sense of Mind. Please be sure to like and subscribe to the YouTube channel and subscribe to the podcast. Also consider giving this show a 5-star rating on whatever platform you use. As always, this channel is brought to you by the Diamond Mind Foundation. This episode was written and produced by me, Andrew Cooper Sansone. Thank you so much for watching. I'll catch you next time.